To understand how populations change through time, we need to look at demographics. Demography is a study of how vital statistics of a population change over time. In particular, we want to understand the relationship between death rates and birth rates, which are how we subtract and add individuals to a population. So one way of resolving how a population can change through time is by constructing a life table. And a life table separates out the members of a population by age to see how they survive from one year to the next. And usually we start by following a cohort or group of individuals of the same age. And we have an example life table using a species of squirrels found in the Sierra Nevada in the western United States, Belding's ground squirrel, which shows a number of things about how the population is constructed. So it shows us the age distribution of males and females. So by taking a census or doing a mark recapture study, we can look at data across different years. So from the 0 to 1, so these are very young Belding's ground squirrels. At the beginning of the year, 337 females were alive, and 349 males, roughly 50-50. So we can look at the proportion that are alive at the start of the year. We can estimate how many deaths occurred during that year and calculate the death rate and calculate how much on average, how much longer that squirrel that survived has to live. So a couple things we can glean from looking at this particular life table. One is that males don't live as long as females. Males all have died out by the age of 6. Females can live as long as 10 years. We can see that the rate of deaths occurring tends to go down, tends to drop from year to year. So if they can survive through the first year, they're likely to make it to the second year, and so on and so forth. And we can take this tabular data and make a graph, a graph called a survivorship curve. So here's some of our Belding's ground squirrels, and here are some researchers collecting them in a live trap and collecting data on them, like how old they are, maybe banding them so they can identify individuals from one year to the next, probably taking a number of other data on them, their mass, things like that. But we can use that life table and create a survivorship curve and see how the population is distributed in age. So taking the data from that life table, we can plot out on a log scale here, so 1, 10, 100, 1,000, how many individuals are surviving from one year to the next. And you can see that the numbers tend to decline in a very linear fashion. So by the time we get to 9 years of age, all the males have already died out before they turn 6. Females tend to live, may live as long as 9 years. But in both cases, it's a fairly long and steady linear decline. So usually we take survivorship curves, such as we observed in the previous slide, and we can classify them into 3 types. Type 1 is when we have a low death rate at the beginning, in the middle of the life, and then at the end we see an increase in death rates. That's what we observe in humans. In the developed world. You tend to be able to survive for a good long while, and then as we age, suddenly the death rate increases. In type 2 organisms, such as in the Belding's ground squirrel, we see a constant death rate, a constant drop in the line. And in type 3 survivorship curves, we see that it's really hard to get through the early years, very high rate of attrition, a very high rate of death for the young, and then once you make it past a certain threshold, your chances of surviving from one year to the next dramatically increase. And there's a lot of gradations between these three types. So here we can see a representative curve. It's idealized, which means these have been all smoothed out. But you can see that in humans, once we are born, there's a good chance we're going to survive for a good long while. And then when we get past about 50% of our maximum lifespan, so 120 years for a human, the death rate increases very quickly. Very quickly. So not very many people get to be at this maximum age. So most of the deaths are occurring when people have led a long life. In Belding's ground squirrels, the rate of death is pretty constant. So it doesn't matter whether you're a 1-year-old squirrel or a 6-year-old squirrel or an 8-year-old squirrel, your chances of surviving from 1 year to the next are pretty much level at any age. Take, for a third example, for type 3, oysters. Oysters produce lots and lots of larvae, and not very many of these larvae survive to the stage where they will start producing a shell. So you see a huge drop-off in numbers of individuals until once they get to this age where they have settled down somewhere and anchored themselves to a substrate and started producing a shell. If they can get to that point, their chances of surviving for a long time thereafter are very good. So in addition to looking at the survivorship, at looking at the death rate, because survivorship and death rate are kind of two ways of saying the same thing, we also want to look at birth rate, or the reproductive rate. So if a species undergoes sexual reproduction, the demographers are going to look mainly at the females, because the females are the ones who are ultimately responsible for producing the progeny, and eggs are a lot more limited than are sperm, male gametes. So females are kind of the limiting factor in terms of reproduction. So ecologists use a number of different approaches to estimate the number of breeding females. An example from our text is one that's used on a number of different species, which is a type of DNA profiling using a region of DNA called microsatellites. So the way it works is we can collect a skin sample from an individual turtle and look at the DNA, collect the DNA, similar to what you did in the lab from that loggerhead turtle, and looking at what are called STR sites, or short tandem repeats, or they're also called microsatellite regions, looking at 14 different gene loci, and the short tandem repeats vary in number from one individual to the next. They tend to be short repeats of trinucleotides or tetranucleotides or dinucleotides, and here they're just showing you with trinucleotides, so you can have 5 copies of TAC, 6 copies of GTG somewhere else in the genome, 4 copies of TTG, and you can look at 14 of these different loci and get a profile which ends up being like a genetic fingerprint. So an individual will only have so many of each of these short tandem repeat loci, and you can compare because they are the numbers of units in each of these repeats is going to vary from individual to individual. So it's just like a fingerprint, only it's encoded in the DNA. And you can use those data from comparing these different females to look at parentage in the egg. So if you collect an egg from the nest, you can see who is the mother by comparing these microsatellite loci. So here, for example, is DNA, the microsatellite profile from an egg shell. So we can look at that and say, okay, how many of this particular orange locus? 6. How many of this darker orange locus? 6. How many of this red locus? 7. And we can say, okay, which one of these females had that same pattern? It looks like it's number 109. So we can say that the mother of this eggshell was breeding female number 109 and not number 108. So we can come up with a different type of table called a reproductive table, looking at how the age of the mother affects her ability to reproduce, or her what we call fecundity. So here is a fecundity table, or a reproductive table, for these Belding's ground squirrels, and you can see that females aged 0 to 1 aren't sexually mature. They don't reproduce. At 1 to 2 years, many of the females are old enough to produce a litter, but their litters tend to be smaller. And being interested in the reproductive capability of that litter, we want to know how many females are in the litter. As we get to 3 years, almost all of the females are producing litters. The litters are a little bit larger than the previous year. And then when we get to the age between 4 and 5, almost all of the females are littering, and their litters are at their maximum size, about 5.4 pups per litter. And then after the age of 5, they'll continue to keep littering, but the size of the litters tends to go down. So their reproductive output is in decline after they've reached what for a squirrel is middle age. So using these data, we can figure out whether or not a population is increasing in size or declining in size, and we come up with different population growth models to explain what we could predict for a population. So one model for looking at population growth is the exponential model. And this occurs in an idealized, unlimited environment. So if you don't have to worry about running out of food or space or water or any of the resources that you need, an organism can grow exponentially. So how do we figure out what the rate of increase is? Well, the change in the population size, how many individuals there are from one year to the next, is going to depend on how many organisms are born, how many immigrants are entering the population, how many organisms die, and how many immigrants leaving the population. So these are positive terms, and these are negative terms. So often, if we've got an enclosed system and we have unlimited resources, we can ignore immigration and emigration. So the growth rate in this population is going to be the birth rate, which is positive, minus the death rate. So as the population grows, we can express the change in the size of the population in a certain unit of time, and the change in time, say 1 year. After 1 year, the number of the change, the delta in a population, is going to be the number of births minus the number of deaths.